channel. Thanks very much for the subscription. For those who subscribe, those who have not yet subscribed, please do one thing: subscribe, put on notification. We continue to learn content with true facts of medicine and health. Today we are going to be looking at the Notch pathway, Notch signaling pathway, and this pathway is very common in multicellular organisms and is involved in regulation of cell proliferation, cell fate, apoptosis, and neurodevelopment. The target genes we have here, we have P1, we have P21, we have cyclic D1 and MYC genes. This pathway has molecules and proteins, as you are going to be finding them, and this is what they stand for in their full forms. We are going to find a protein called ADAM, and this one is going to be a disintegrin, and the metalloprotease. BJ is DNA binding protein. NICD is going to be the Notch intracellular domain. Then NECD is Notch extracellular domain. TM is transmembrane protein. Then this one is going to be mastermind like activator 1. Then this one is the mind bomb. So we are going to be coming across these short forms in our pathways, and this is what they stand for. Let us go to the pathway and we see what we are going to have. Two cells. We are going to have a signal sending cell. Then down here we are going to have a signal receiving the cell. So what is going here? We have our delta ligand. This is our delta ligand with our mind bomb attached on it. So this is the delta ligand. So this mind bomb is going to activate the delta ligand by ubiquitinating it. So by ubiquitinating it, it's going to be active now. So once it is activated on our signal receiving cell, we have our molecules. So this is the NIC and the Notch intracellular. This is the Notch intracellular domain. This is the Notch extracellular domain. Then this one is the transmembrane protein. So what is going to happen? We are going to have this active delta ligand complexing. It's going to complex with the Notch extracellular domain. Once they complex, we are going to have our membrane bound protein here. And this is an enzyme. this enzyme is called TACE. So this TACE is going to cleave the NECD delta complex. This is referred to as S2 cleavage. So after S2 cleavage occurring, we are going to remain with the transmembrane protein and the notch intracellular domain. Then we are going to have a complex of alpha secretase complex. So this complex of enzymes is going to come and cleave the TMNICD complex. After cleaving, and this one we refer to as S3 cleavage. After that cleavage, we are going to have the notch intracellular domain being released inside the cell. So as it is released inside the cell, it's going to translocate into the nucleus. In our nucleus, we have our DNA, and this DNA is bound by a co-repressor. So as this molecule translocates into the nucleus, this co-repressor is going to be replaced by this molecule NICD binding to, so we are going to have NICD binding to our RP BG, which we saw as the DNA binding protein. Then remember, we had our mastermind-like activator is also going to come here. So this complex is going to lead to what we call transcription. This complex is going to be degraded in the proteasome.